Hi everybody, my name is Nour Boura. I am your colleague in this class. I will talk about an interesting topic under the title of DNA fingerprinting. First, let's talk about what does DNA fingerprinting mean. DNA fingerprinting is a technique used by scientists to distinguish between individuals of the same species, using only samples of their DNA. The only difference between people is the order of the base pairs. Who invented this incredible technique? The process of DNA fingerprinting was invented by Alec Jeffreys at the University of Leicester in 1985. Now let's talk about the stages of DNA fingerprinting. Stage 1. Cells are broken down to release DNA. If only a small amount of DNA is available, it can be amplified using the polymerase chain reaction, which is known as PCR. Step 2. The DNA is cut into fragments using restriction enzymes. Each restriction enzyme cuts DNA at a specific base sequence. The sections of DNA that are cut out are called restriction fragments. This yields thousands of restriction fragments of all different sizes because the base sequences being cut may be far apart or close together. Stage 3. Fragments are separated on the basis of size using a process called gel electrophoresis. DNA fragments are injected into wells and an electric current is applied along the gel. DNA is negatively charged, so it's attracted to the positive end of the gel. The shorter DNA fragments move faster than the longer fragments. DNA is separated on basis of size. A radioactive material is added which combines with the DNA fragments to produce a fluorescent image. A photographic copy of the DNA bands is obtained. Stage 4. The pattern of fragment distribution is then analyzed. This is a sample picture of DNA fingerprinting. Finally, let's summarize the benefits of DNA fingerprinting. DNA fingerprinting is a way of telling individuals of the same species apart. DNA sequences are variable and can therefore be used as identifying characteristics. DNA fingerprinting has advantages over other sources of evidence. It can highly accurate and can be gathered from trace crime scene evidence. I hope you were interested in this simple presentation about DNA fingerprinting. Thank you for listening.